Hello everyone. Let's get started. Today we're going to be talking about mutations. In lecture we've been discussing DNA and RNA and the basic processes of DNA replication and gene expression. As we move on to considering the problem of mutations, it's important that we remind ourselves of the levels of structure of the DNA molecule. Remember, in living cells, DNA exists as a double-stranded molecule, but we can describe it as having multiple different levels of structure. The primary or the simplest level of structure that you can describe DNA as having is the order of the bases in the molecule. In other words, the specific sequence of the A's and the T's and the G's and the C's. That's the simplest way to describe a piece of DNA in its primary structure. The secondary structure of DNA, then, is the next level of complexity of the molecule. The two strands of DNA that come together and complementary base pair with each other doesn't stay as a flat two-dimensional structure. Instead, it creates what we call a double helix. On this slide over on the right, you're looking at a diagram of how we would perhaps draw a double-stranded piece of DNA, indicating the specific sequence of the base pairs, and how that molecule would actually exist in nature, which is to form this double helix. Primary structure, secondary structure. The tertiary structure of DNA is then the next level of complexity that this molecule takes in living cells. And that can be either the form known as chromatin or the chromosome. Chromosomes are actually interesting structures because technically they only occur in a cell at a very particular time in its life cycle. And that is when the cell is preparing to divide. Most of the time, the DNA molecule exists as chromatin. So all chromatin is this DNA molecule, this double helix, associated with what are called histone proteins. Now, histone proteins only exist in eukaryotic organisms. The bacteria and the archaea actually have what are called histone-like proteins. They're very similar to histone proteins, but they're not exactly the same. The double-stranded DNA molecule will wrap around these histone or histone-like proteins in a very ordered way. These bundles of DNA and histone protein will then stack up in a very complex structure that is referred to as being compacted DNA. That is what chromatin is. It's DNA that has been systematically wrapped around histone proteins and compacted. Now, when it comes time for the cell to divide, the chromatin is going to become even more folded, even more condensed. And that's what allows these chromosomes to form. Remember, when a cell divides, it has to divide everything inside of itself into two brand new cells. And that includes the DNA molecule. The DNA has to be copied or replicated, and then those two copies have to be separated out and sent to each of the two new daughter cells. In order to make that process as efficient as possible, the DNA in the chromatin form assumes the chromosome form so that the chromosomes can be physically moved to different parts of the cell. Chromosomes are simply DNA that has been wrapped around histone or histone-like proteins, compacted, in other words, and condensed. That's the terminology we use. Chromosomes are therefore more folded and pressed into these complex three-dimensional structures than chromatin is. Down here at the bottom of the slide, you're looking at a drawing of a chromosome over here on the left and a replicated chromosome over here on the right. This chromosome has gone through the process of DNA replication, so now there's a copy sitting next to it. We often draw chromosomes as replicated chromosomes, having this sort of an X shape. The two copies are attached to each other. We refer to them as sister chromatids. On this slide, you're looking at a drawing that shows this progression of the DNA molecule from its secondary structure, again, the double helix, into chromatin, which is wrapped around histone proteins. Now, what they're showing you in this drawing is that the DNA wraps very precisely. It wraps twice around each of these cores of histone proteins, or histone-like proteins in the prokaryotes. This structure right here, the combined DNA and histone proteins, is referred to as a nucleosome. Nucleosomes are basically the subunit of chromatin. All of these nucleosomes are then carefully folded, carefully stacked together to create this very complex molecule that we call chromatin. Now, within the DNA molecule, there are regions that are referred to as genes. Remember what a gene is. A gene is a piece of DNA in its, primary sequence, its primary structure. It's a sequence of bases, and that those particular bases contain code. Now, most people will tell you, oh yeah, a gene is a piece of DNA that codes for a protein. And that's correct, but that's an incomplete definition, because genes also contain a second type of code within them. They have code for building a protein, and they also have regulatory code, or a sequence of bases that has a purely regulatory function. The regulatory code in a gene is the code that determines when the protein will be made, in fact, if the protein will be made, when it will be made, and how much of it will be made. So every gene has regulatory sequence in it, regulatory bases, and protein coding bases. Now I have a star next to the word protein here, because the other thing we need to know is that in eukaryotes we say one gene, one protein. In other words, each individual gene codes for a single protein. But that's not the case in prokaryotes. In prokaryotes, sometimes one gene actually contains the code to build multiple proteins. We refer to these types of genes as operons. You discuss operons at length in genetics. The important thing to remember, though, is that in both prokaryotes and eukaryotes, every gene has both protein coding sequence in it and sequence that will regulate if, when, and how much of that protein will be made. Now, humans and other diploid organisms inherit two copies of every gene in the DNA molecule. Now, The other term we need to apply here is this term allele. Remember what an allele is. It's a particular version of a gene. So while a gene codes for a particular protein, there can be different versions of that particular protein. And that's the individual forms of that gene are referred to as alleles. So you and I, as diploid humans, we inherit 2 copies of every allele. We inherit 1 copy from our mother and 1 copy from our father. Other terminology to review are the terms genome, genotype, and phenotype. When I use the term genome, when you read the term genome, we're talking about the collection of DNA that an organism has. The genome includes all the DNA, not just the genes or the alleles within the DNA, but all of the DNA. Because, of course, there's a lot of DNA that is not genes. The genotype of an organism is that organism's collection of alleles. Remember, you and I, as humans, have the same genes in our DNA, but your alleles for those genes might be different from my alleles. So your genotype is different from my genotype. And finally, this term phenotype. This term is defined as an organism's collection of traits. Now, sometimes students want to think that the phenotype is the collection of physical traits, how the organism looks or is structured. But remember, your phenotype is all of your traits: physical traits, chemical traits, biochemical traits, behavioral traits, every single thing that makes you a unique organism. We now know the primary structure of human DNA, don't we? Many years ago now, the Human Genome Project was set up and funded to help us determine the exact sequence of base pairs in the human DNA molecule. If you're not aware, that project showed us that there are approximately 3 billion base pairs in human DNA. It's an enormously long, complex molecule. Now, just because we know the primary structure, just because we know the sequence of all the bases, does not mean that we know all of the genes yet, and we don't. We're finding new genes all the time. The current estimate is somewhere close to about 30,000. The current estimate is that human DNA contains about 30,000 genes. Someday we will know exactly how many genes are in human DNA, where they are, and what protein they code for. But we don't know that yet. Many other genomes have also been sequenced. In other words, we know the primary structure of many other genomes, many other organisms' genomes. We have a public database of all this genome sequence called the NCBI, the National Center for Biotechnology Information. This is a database that is funded by the US government. It is free and open to everyone. It stores all of the known sequence data, all of the gene sequence data, all of the sequence of amino acids in proteins that we know about. And again, it's free to use. So students and researchers and anyone who's interested can access the NCBI. Remember, all of the living things on this planet can be organized into one of 3 groups: the bacteria, the archaea, and the eukarya. These are what we refer to as the domains of living things. The bacteria evolved first, followed by the archaea, followed by the eukarya. We said that bacteria and archaea are considered prokaryotes. They have 1 chromosome inside their cell. There are a couple of exceptions to that, but most prokaryotic organisms have one circular chromosome. That's their genome. They also have extra chromosomal DNA, however, called plasmids. Remember, the DNA that's found in plasmids is not necessary for that organism's survival. It's extra DNA. It gives the organism the ability to make proteins that are extra, that often give that organism a survival advantage. Remember, things like antibiotic resistance are usually located on plasmids. Eukaryotes are very different. Eukaryotes have multiple linear chromosomes. Interestingly, though, eukaryotes also have extra chromosomal DNA that we sometimes forget about. They have a small amount of DNA in their mitochondria. They have a small amount of DNA in their chloroplasts if they are photosynthetic. And occasionally, eukaryotic cells will contain plasmid DNA. Some of the lower single-celled eukaryotes, particularly, are known to carry plasmids. So it doesn't matter if it's a prokaryotic organism or a eukaryotic organism. It's got its central genome, its molecule of DNA that contains all the genes that are necessary for it to survive. And it also contains some extra chromosomal DNA. On this slide, you can see a drawing of a bacterial cell, and the artist is showing us that there's going to be a genome in here. There's going to be a single circular chromosome in here. And there's also sometimes going to be extra chromosomal DNA. Remember, these plasmids are kind of randomly distributed in bacterial populations. Some bacterial cells have one plasmid, some have multiple plasmids, some don't have any. Down here, you're looking at an actual plasmid underneath an electron microscope. Again, it's a circular piece of DNA. It does contain genes, but these genes are not required for the survival of the organism. On this slide, we're looking at a picture that's reminding us that prokaryotic organisms store their DNA right in the cytoplasm because they don't have any organelles. This represents a bacterial or an archaeal cell. You can see the cell wall and the cell membrane. You can see the cytoplasm in this yellow gold color. There are little red dots in here that represent ribosomes. And here's this large mass called the nucleoid in green. This is the chromatin. This is the DNA molecule that has been compacted with histone-like proteins. DNA is not simply floating around in the cytoplasm exposed all the time. Instead, it's associated with protein. Over here, you're looking at a eukaryotic cell. Remember, eukaryotic cells have organelles in them, including a nucleus, and this is where the DNA is housed. It's chromatin at all times, except for when the cell is preparing to divide. At that time, the chromatin will become compacted and condensed, and chromosomes will form.